in addition to the notch signaling, there are other signals that can alter the positional value or identity of a location in the brain. So now we're looking at mouse. So we're jumping back and forth. Retinoic acid is a derivative of vitamin A, and it's a really strong mutagen. And so if you use it in the laboratory, it tells you on the bottle, be careful, this has been shown to cause massive changes in development in animals. So, for example, if you expose vertebrate embryos to high doses of retinoic acid early, you get homeotic transformation of the vertebrate. So homeotic changes or homeotic genes we discussed when we talked about Drosophila. They're the ones that change the different parts of the body to another, right? The Hox genes. So homeotic transformation is also a transformation of basically location, and retinoic acid can induce that. It is highly diffusible, so it's not a membrane-bound protein, so it can diffuse across plasma membranes and go directly into the nucleus and act as a transcription factor. And it's known now that retinoic acid interferes with normal expression of Hox genes. And so here we see the developing somites, so they're labeled S1 through 10. And if you look at the expression levels of retinoic acid in the different areas of the mouse, we see where there is hardly any retinoic acid, or so you see gradients of retinoic acid being expressed, so that it's higher here and lower here, it's higher here and lower here. So it's thought that changes in the concentration of retinoic acid help to dictate the location of the different parts of the body along the anterior-posterior axis as it's developing. And we'll also talk about this FGF gradient later. Okay, so this is another drastic picture to show you how retinoic acid affects development in these vertebrate animals. So this is the wild type mouse. Right? It's got all its vertebrae, it's properly developed. This mouse got exposed to an intermediate dose of retinoic acid. You can see its vertebrae and its bone formation is not normal. It's shorter and some of the vertebrae are fused. And then this mouse, and I'm sure it did not live, but they probably got exposed to even more and just stopped forming more posterior structures because of the high doses of retinoic acid. So the discovery of these changes in response to retinoic acid was and how they regulate expression of Hox genes was was awarded the Nobel Prize in 1985. So let's take a break, and then we'll talk about Hox genes and their positional identity in the vertebrate embryo. So it's 3:50, so let's come back at 3:55. I don't think they knew.
Let's see what happens.
You're
Oh
just
my
gonna
God.
toss this stuff on the side.
Yeah, you can't do things just like that.
Oh.
I think it was discovered first by accident.
stuff like that, it doesn't affect the mother? Or how is it that they?
So I think they either, yeah, it probably didn't affect the mother. I don't know how they did it. I didn't, they probably either injected like into the, probably the
That seems like,
expose them early. Yeah. They gave high doses of vitamin A to the mothers on day 8 of pregnancy. It's early.
so they feed with them?
So they gave high doses of retinoic acid to the mothers on day 2. How does it affect the mother?
Wait, does that mean that they fed it to them or they injected them?
I'm not sure if they.
Okay, but somehow they got it.
Yeah, so here I'm looking at the Nobel Prize website right now, which is where I got this. And so it's saying, so 2 of the 3 mouse embryos show malformations caused by high doses of vitamin A, which was given to the mothers on day 8 of pregnancy. And then I thought this is interesting. It's well known that high doses of vitamin A taken by women early during pregnancy can disturb the regulation of Hox genes, either in women and or can cause severe malformations.
So don't take a lot of vitamin.
No.
Right.
It's good to know. Carrots. But I don't think they have it in,
Maybe, it seemed like.
they don't have it in like the dose that, you know.
It just says something like that.
Yeah.
These are retinoic acid, not iodine.
Oh, you put the pink one in? Where's the pink one go? Oh, just the-
Yeah, I'm
Maybe I
sure
covered
there's
up.
not,
Like
you
the
don't need
yellows.
very much to do what
I don't know.
I wonder if there's anything about milk or something. Don't have vitamin, yeah, they don't have vitamin E and vision hormone. I think it's like retinoic acid, also intermediate for other metabolites.
Well, we have someone coming. The 12th, right? Yeah,
Okay.
12th.
Okay, so let's pick up, and then I'll be caught up. I think I'll be caught up on my. Okay, so we're talking about Hox genes in vertebrates, and so this is a cartoon that kind of demonstrates how Hox genes are thought to work. So if you have here, there's just 4 different genes, and their expression in the anterior versus posterior part of the body may be different. So for example, if gene 1 is expressed throughout the body, and in gene 2 it's expressed just from